Hello students. Today we start with our metabolic reactions. Now carbohydrate metabolic reactions. So what is this metabolism? Before that we can just see that after the digestion we had a glucose as a after the digestion we had a glucose as our product. So this glucose now goes to the cells. Galactose and fructose are also produced by digestion, but these two go to the liver and they are converted into glucose. So now the glucose metabolism is the main metabolism where glucose is converted to pyruvate. This is the first metabolism. It takes place in all the cells and this is known as glycolysis. So the glycolysis is what? It is the breakdown of this glucose into pyruvate, two molecules of pyruvate with releasing lot of energy. So it is the major pathway. So now today we will deal with the glycolysis. Even if I have already loaded the videos about general carbohydrate chemistry, so if you have any problem then you can see that videos also. So now today we start with glycolysis. So glycolysis, what is glycolysis? It is the oxidation of glucose or glycogen to pyruvate or lactate, and that is known as glycolysis. Glyco means glucose, lysis is breakdown. So it is the breakdown of glucose in presence of oxygen, then it is known as glycolysis. And the product what we have, product is pyruvate in case of aerobic glycolysis. In case of anaerobic it is lactate. So it is also known as Embden-Meyerhof pathway, EM pathway. It is also known as EM pathway, that is Embden-Meyerhof pathway. It occurs in all tissues. Even erythrocytes and nervous tissue derive its energy mainly from glycolysis. So without glycolysis, this RBC cells and nerve cells, brain is impossible to survive. So they need energy that comes from glycolysis. So glycolysis is important pathway as it gives energy to all the cells. It can utilize oxygen and also function in absence of oxygen. This pathway is otherwise also important because it in presence of oxygen it can occur and also in absence of oxygen, that is aerobic also, and in anaerobic condition also this glycolysis can take place. So there are two phases, aerobic phase and anaerobic phase. Now we see the biomedical importance of glycolysis. Why this glycolysis is important for us? This pathway is meant for provision of energy. Hence we have seen that brain cells, RBC cell, each and every cell gets its energy from this pathway only. It has importance in skeletal muscle. In skeletal muscle it plays an important role because there it takes place in anaerobic condition also, as glycolysis provide ATP even in absence of oxygen, that is anaerobic condition. When we do lot of exercises, then our muscle cells does not get that much oxygen how much it requires. So in anoxic episode it can help to survive in that conditions, that is this anaerobic glycolysis occurs in muscle. So for muscle it is very important. Then for heart muscle, as compared to the skeletal muscle, heart muscle is adapted for aerobic performance. It has relatively poor glycolytic activity and poor survival under the condition of ischemia. Now role in cancer therapy. In fast growing cancer cell, rate of glycolysis is high. So cancerous cells, they need energy and metabolism, that's why they increase glycolysis rate and produce more and more pyruvate than TCA can handle. So the pyruvate is increased here. How much the TCA can reuse, TCA can use, instead of that too much pyruvate is produced because of cancer cells. There are many cells which produce which increases the rate of glycolysis, and rate of pyruvate production is too much. So accumulation of pyruvate thus in the cells lead the lactic acid formation, and this lactic acid makes it acidosis. So it creates an acidic environment which is congenital for cancer therapy. So that is and congenital for cancer therapy. Now next, it plays important role in hemolytic anemias. Inherited enzyme deficiency like hexokinase. So deficiency of hexokinase and pyruvate kinase deficiency and can produce hemolytic anemia. The cell can produce hemolytic anemia. So RBC cell without this glycolysis will be damaged or get hemolyte. So it may give production of hemolytic anemias. So we will have hemolytic anemia as RBC cell cannot survive without this glycolysis. So this is the only pathway which gives its energy. Now the next important is it plays a sole pathway provision for energy for RBC and lens cell. So lens in the eye also get energy from this pathway only. The next importance it is that it is linked to many other pathways. So this glycolysis has been a link to other pathways like glucose 6 phosphate produced in this pathway is intermediate in pentose phosphate pathway, that is HMP shunt. It is also in uronic acid pathway with dihydroxyacetone phosphate, which is used in triacyl glycerol synthesis, phospholipid synthesis. This dihydroxyacetone phosphate, it is intermediate in glycolysis. We will see now when we will see the pathway. 1,3-biphosphoglycerate, 2,3-biphosphoglycerate, these are also intermediate in RBC cells. Then pyruvate is also an intermediate for the production of alanine. So in this way we have seen that this glycolysis have too many importance. So now it is necessary to read or to learn pathway. Now types of glycolysis. So we have seen that glycolysis are of two types: aerobic glycolysis and anaerobic glycolysis. Aerobic, that is in presence of oxygen. Anaerobic, that is absence or less of oxygen. It occurs in all cells. Aerobic glycolysis occurs in all cells, but anaerobic occurs in muscles, kidney cells where mitochondria is absent in that cells. The end product of aerobic glycolysis, it is pyruvate, and anaerobic glycolysis, it is lactate. Now net gain of energy, it is 8 ATP. Before it was 8 ATP, but now it is 7 ATP as NADPH produces 2.5 ATP only. So it has a net gain of 7 ATP, while anaerobic glycolysis produces 2 ATP only. Now we start with the glycolysis steps. So glycolysis, it occurs in 10 steps. First step is phosphorylation of glucose. So in the first step, the pathway starts from glucose. So the glucose is phosphorylated. Phosphorylated means addition of phosphate takes place. So this glucose, on the 6th carbon of this glucose, it gets phosphorylated and phosphate is added to the 6th carbon. So if this phosphate is added, so somebody is to be there who donates this phosphate. So here in this reaction, phosphate comes from what? ATP, that is adenosine triphosphate. This ATP has adenosine with three phosphates, triphosphate. So one of the phosphate is donated to this glucose, and glucose is now converted into glucose 6 phosphate, and ATP is now converted into ADP, that is adenosine diphosphate. This reaction takes place in presence of hexokinase or glucokinase enzyme. Hexokinase is a non-specific enzyme. It is present in all cells. But glucokinase is present only in liver cells. So it is specific enzyme. it acts only on glucose, whereas hexokinase acts on hexose sugar. This ATP, this hexokinase or kinase enzyme always it has a cofactor or coenzyme, that is magnesium ions. So metal ions, magnesium are essential for this reaction. So in the first step, so what we say, that glucose was phosphorylated. The reaction is catalyzed by glucokinase in liver cell and non-specific hexokinase in liver and extrahepatic tissue. The reaction is irreversible, so we have only one-sided arrow. So glucose 6-phosphate is then not converted into glucose. It is only irreversible reaction, and we have seen that it is in presence of magnesium ions. Now difference between hexokinase and glucokinase. So hexokinase, we have told you that it is non-specific, that is it acts on other hexose sugar also. But glucokinase is specific, it acts only on glucose. Then it is more stable, hexokinase is more stable, whereas glucokinase is more labile. It is not stable. So it is inhibited by glucose 6-phosphate. It is not inhibited by glucose 6-phosphate. It has no change with glucose feeding. If glucose is added, it does not change, but increase of feeding of glucose after fasting. It increases feeding of glucose after fasting. As it is fasting, then glucose starts feeding. It is inhibited by glucocorticoids and growth hormone, and insulin has no effect on hexokinase. It is inhibited by glucocorticoids and growth hormone, but here glucose and insulin stimulates this pathway. So glucokinase enzyme is stimulated by glucose or insulin. So synthesis is induced by insulin. Main function is to make available glucose to tissue for oxidation at low blood glucose. The main function of this glucokinase is to produce or is to clear glucose from blood after meals and blood level greater than 100 milligram per deciliter. Now number 2 step. First step, it was phosphorylation, where glucose is phosphorylated with glucose 6-phosphate at the 6th position. Now in the second, it is isomerization. What is that? Isomerization of glucose 6-phosphate. So this glucose 6-phosphate, so it will be converted into its isomeric form, that is fructose. So this glucose is converted into fructose with 1 phosphate, that's why it is known as fructose 6-phosphate, and the enzyme is phosphohexose isomerase. So here is isomerization reaction, so enzyme is isomer. Then phosphorylation of fructose 6-phosphate. Now the first step is repeated. First step, it was phosphorylation of glucose. Now here, after the conversion of fructose, now one more phosphate is added and the molecule become fructose 1,6-biphosphate. So before that it was having only 1 phosphate. Now one more phosphate is added. That fructose, it was 6-phosphate. Now at the first position it has another phosphate. So the compound formed is fructose 1,6-biphosphate. The enzyme is phosphofructokinase. And you see here first phosphate was there. Now at the first position you have another phosphate. That phosphate is also donated by ATP. So we have seen that first and third reaction. This is the third reaction. ATP are utilized. Here ATP are utilized. No synthesis of ATP. Only they are utilized. So we will see that how to learn enzymes. So enzymes we can learn like this: when there is exchange of ATP or ADP, when in reaction there is ATP involved, then the enzyme is kinase. Always the enzyme is kinase. For that kinase we use what is this? Product name. So this is phosphofructokinase. Understood? In the first reaction it was glucokinase. So okay, now it is phosphofructokinase. That's why the kinase name, when ATP ADP are exchanged, and always kinase is working in presence of magnesium ions. So you remember, so whenever it is kinase enzyme, magnesium ions are essential and ATP is involved. So it is mostly phosphorylation reaction, and these phosphorylation reactions are always irreversible reaction. Now the fourth step is cleavage of fructose 1,6 biphosphate. So now the fructose 1,6 biphosphate, it had two phosphate at first carbon and at sixth carbon. So these two phosphates are now cleaved or broken down into two 3,3 carbon sugars. So fructose is 6 carbon sugar. So this 6 carbon sugar is converted into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Both are having 3 carbon. One is keto compound and one is aldehyde compound, and both the phosphates are also shared, one with dihydroxyacetone phosphate and the other is glyceraldehyde 3 phosphate. And the enzyme here responsible is aldolase, and this is aldolase A. So the enzyme is aldolase A, which breaks this 6 carbon into 3,3 carbon 2 compounds, that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Now the fifth step is again isomerization. Second step was isomerization. Now the fifth step is also isomerization, where we see the isomerization of dihydroxyacetone phosphate. So dihydroxyacetone phosphate is converted into glyceraldehyde 3 phosphate. So this enzyme responsible for here is triose phosphate isomerase. So when there is isomerization reaction, the enzyme is isomerase, and here we write the name of both. So triose phosphate isomerase. So dihydroxyacetone phosphate, that is keto compound, 3 carbon keto compound, is converted into 3 carbon aldehyde compound. Then comes the sixth step. So in the sixth step, this is very important step, as here glyceraldehyde 3 phosphate. Now we have seen that glyceraldehyde 1 glyceraldehyde was formed, and the second is that dihydroxyacetone phosphate was converted into glyceraldehyde. So now we have 2 glyceraldehyde molecules. So this glyceraldehyde 3 phosphate, it is converted into high energic 1,3 biphosphoglycerate. So it has 2 phosphate on 1 and 3 carbon, and that's why it is 1,3 biphosphoglycerate. This phosphoglycerate is reduced. You have seen that H ions are here. H ions are donated. You can see 1 H from here and 1 H from here is donated. Therefore this NAD comes here. It takes away the 2 hydrogen molecules. So these are reducing equivalents, which takes the hydrogen which are donated by this and compound, and that's why they are known as reducing equivalent. This NADH, the full form of NADH is nicotinamide adenine dinucleotides, and these 2 protons are there. This NAD, it now enters, this NAD produced here, it now enters the mitochondria and will run electron transport chain, and this NADH will be converted into 2.5 ATP. So it is, that's why it is known as oxidative phosphorylation. So this reaction is very important. It is the first oxidative phosphorylation reaction of glycolysis. So in the conversion of glyceraldehyde 3 phosphate to glycerate is in the presence, and the enzyme is dehydrogenase, because 2 hydrogen are removed. That's why this is known as dehydrogenase enzyme. So the enzyme where we see that NADH, FADH, FMNH, all these compounds are formed, then that time the enzyme is always dehydrogenase, and dehydrogenase enzyme is always opposite to kinase. That is, in kinase we are writing the name of the product, but here we write the name of reactant. So the enzyme here is glyceraldehyde 3-phosphate dehydrogenase. So glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate in presence of enzyme glyceraldehyde 3-phosphate. Here NADH is produced. This NADH then goes into the electron transport chain in mitochondria and produces 2.5 ATP. Now, and in the past it was 3 ATP. So here two important mechanism occurs, that NADH synthesis occurs, which goes into the mitochondria and is the oxidative phosphorylation, where there it produces 2.5 ATP. And you also know this NADH what produced here is utilized when pyruvate is converted into lactate. That is an anaerobic reaction. So that oxidation, the conversion of pyruvate to lactate will utilize this NADH. So these NADH are balanced over there. So therefore in anaerobic glycolysis we have only 2 ATP net gain. Now production of ATP from 1,3-biphosphoglycerate. So this 1,3-biphosphoglycerate is now high energy compound. This high energy compound is converted into 3-phosphoglycerate, which is reversible reaction. And you see here is also a kinase enzyme, because here it is also an exchange of ATP ADP, but here in first and third reaction we saw ATP was utilized, but here ATP is synthesized. This high energy product, reactant produces ATP, and therefore the enzyme is kinase. In the kinase enzyme, we can write phosphoglycerokinase is also right, biphosphoglycerate kinase is also right. It is the biphosphoglycerate kinase, or we can also write phosphoglycerokinase. It utilizes magnesium ion also and produces ATP. This reaction is opposite to the sixth reaction. That was oxidative phosphorylation, where ATP was produced in electron transport chain. But here this is known as substrate level phosphorylation. Why it is substrate level? Because from the substrate only the ATP is produced here only. So no need to go in mitochondria or electron transport chain. Now we come to the 8th step. It is the shift of the phosphate group from carbon 3 to carbon 2. Now the phosphate group which was on the third number of carbon, it is now transferred to 2 number of carbon. And this change, where we see change the position of a substance is there and a substrate is there, then there the enzyme comes is known as mutase enzyme, mutase or epimerase. In this way we can learn the enzymes. When there is exchange of position, then the enzyme is mutase or epimerase. Hence here the enzyme is that phosphoglyceromutase enzyme. So 3-phosphoglycerate is now converted into 2-phosphoglycerate. Now the 9th reaction, where dehydration of 2-phosphoglycerate takes place. So this 2-phosphoglycerate is now dehydrated. So water is removed and it is converted into phosphoenol pyruvate. So this phosphoenol pyruvate, here the enol compound is formed, therefore the enzyme is enolase. And very important, this enzyme in blood collection, है ना? When you estimate blood glucose, then the vials what we use is fluoride vials. Why we use fluoride vials there? Because this enolase activity is stopped, inhibited by fluoride ions. So these fluorides, so when we collect the glucose RBC, in RBC glycolysis will take place. But if we take this in fluoride vials, if we take the sample in fluoride vial, then glycolysis will not appear and all the glucose will remain as it is. It won't be converted into pyruvate. When if the technician does the estimation maybe after 2 hours or 3 hours, then also the sugar will remain static. So for that's why we carry this, collect this blood sample in fluoride vials. The fluoride vials are purple, what we say, gray color vials. Cork of that vial is gray colored. Now next we come to the 10th step, that is the formation of pyruvate producing ATP. So last product, that is aerobic glycolysis ends here, where the end product is pyruvate. So this phosphoenol pyruvate has one phosphate, but here there is no phosphate. So this one phosphate is released. So there has to be somebody to take the phosphate. So this ADP, that is adenosine diphosphate, it takes one phosphate and is converted into ATP in this way, and the enzyme here is pyruvate kinase. So here also we will write magnesium ions. So here kinase enzyme is there, and we have seen that kinase enzyme is always in the presence of magnesium ions. Here I have missed writing, so you take care that phosphoenol pyruvate is converted into pyruvate and one ATP is generated, and enzyme is pyruvate kinase. And this is also irreversible. This is third irreversible reaction. So we have seen all the kinase reactions are irreversible. Exceptional case was only up to seventh reaction, that was phosphoglycerokinase. That's irreversible reaction. That was reversible reaction. Then rest of the three, first reaction, third reaction and the 10th reaction, are irreversible reaction. Now anaerobic glycolysis does not end till pyruvate. Anaerobic glycolysis produces lactate. So there is 11th reaction also. The 11th reaction is conversion of pyruvate into lactate. So here you see, this is the reaction, reverse reaction. Here the hydrogen ions are taken by the pyruvate. So this NADH is the donor and it gets converted into NAD. So in the sixth reaction, where NAD was converted into NADH, it is just the reverse, and that NAD that was produced there is cancelled here. So anaerobic glycolysis, the final product is lactate and the enzyme is dehydrogenase. Of course it is NADH, so that's why it is dehydrogenase. And in dehydrogenase, this is the exceptional case where the product name is added, that is lactate dehydrogenase, because pyruvate dehydrogenase we also have when pyruvate is converted into acetyl coenzyme. So here it is lactate dehydrogenase, and the reaction is reversible reaction. So lactate is also converted into pyruvate and then enzyme is lactate dehydrogenase. Now the complete pathway we can see. Here are 10 steps. So in the first step we saw glucose was phosphorylated into glucose 6-phosphate. Then this glucose was isomerized into fructose 6-phosphate. This fructose 6-phosphate now had one more phosphate or phosphorylation and converted into 2 phosphates, that is fructose 1,6 biphosphate. Now this fructose 1,6 biphosphate was broken down into 2,3,3 carbon compounds, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate does not proceed further, so it is again converted into glyceraldehyde 3-phosphate. So now we have 2 molecules of glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is now dehydrogenated and forms 1,3-biphosphoglycerate. This 1,3-biphosphoglycerate is high energy compound. It produces ATP and it gets converted into 3-phosphoglycerate. So 1,3-biphosphoglycerate, so one phosphate is removed and is converted into 3-phosphoglycerate. Then this 3-phosphoglycerate is converted, change its position from 3 to 2 position. So now it is 2-phosphoglycerate. Now this 2-phosphoglycerate is converted into phosphoenol pyruvate, where it is enolase enzyme and is inhibited by fluoride. Then enol pyruvate is converted into pyruvate, and the enzyme is pyruvate kinase. This pyruvate is the end product for aerobic oxidation. So this pyruvate now goes to TCA cycle. But if it is anaerobic respiration, anaerobic glycolysis, then this pyruvate is converted to lactate, and the NADH produced here is used over here. In this way, anaerobiosis uses this, and this NADH then enters the mitochondrial or respiratory chain, and it produces 380. It was before. Nowadays it is latest version, it is 2.5 ATP produced. Now we see energy production. So how many ATPs are produced during glycolysis? So in hexokinase, first and third step, so both ATP are utilized. So we 1 ATP are minus. Then in the fifth step, NADH is produced, and which is 2.5 into 2, that is 5 ATP are produced. Why it is 2? Because from this glyceraldehyde 3-phosphate, 2 glyceraldehyde molecules are there. Understood? That's why it is 2 into 2 we have done, and now we have 5. In the sixth, 1 ATP is produced. So here also 2 products will be formed, so it will be into 2, and now we have 2 ATP. In last step, pyruvate kinase, also 1 ATP is produced, and here also 2 pyruvate are formed. Therefore the ATP is multiplied by 2, therefore it is 2. So when we total, then 5, 2, 7, 8, 9 ATP, and 2 we will minus, therefore it is total 7 ATP. So in the aerobic condition, 7 ATP are produced. Now in anaerobic condition, we see hexokinase 1 minus, third step also 1 minus, and at the sixth step 2 ATP, and at the ninth step 2 ATP, because that NADP is balanced by last 11th step. So we have only 4 ATP and 2 are utilized. So we have net gain of only 2 ATP. Now inhibitors. In glycolysis we have 3 inhibitors. If arsenate, it inhibits enzyme glyceraldehyde 3-phosphate dehydrogenase. It competes with inorganic phosphate and no ATP is produced. Iodoacetate or iodoacetamide, it binds covalently with SH group, that SH group of glyceraldehyde 3-phosphate dehydrogenase. So both these enzymes are inhibited by arsenate and iodoacetate. So which glyceraldehyde 3-phosphate dehydrogenase enzyme. And the fluoride, I have told you that fluoride inhibits enolase enzyme. So it has its role in the blood collection or glucose estimation. Now clinical importance of sodium fluoride. Why we use sodium fluoride is used with potassium oxalate for collection of blood glucose estimation. The potassium oxalate is used alone, then in vitro glycolysis will reduce the glucose and value in the sample. So what are the functions of fluoride? It inhibits enolase enzyme, it also acts as anticoagulant, and it is also antiseptic. Now regulation. This pathway is working, is going in all the cells, but how it is regulated? So it is regulated by 3 glycolytic enzyme. So that are what? Hexokinase, the first step, third step phosphofructokinase, and last step kinase. So all the 3 kinase enzymes are regulating this pathway. Feedback inhibition, it's okay when induction with insulin, it's glucokinase, or when pyruvate is produced in large, then also it will inhibit. Then covalent modification, we can see that inhibitor is glucagon and activator is insulin. Insulin activates this pathway and glucagon inactivates this pathway. Allosteric regulation, we see that activators are fructose 1,6 biphosphate and AMP, whereas inhibitors are ATP and citrate. Now we can see the pathway like this, the glucagon, how it inhibits. So when glucagon comes, adenyl cyclase is converted into cyclic AMP. This cyclic AMP, active protein kinase, it converts this cyclic AMP is converting protein kinase into active. This active protein kinase will convert active pyruvate kinase into inactive. So pyruvate kinase becomes inactive, so the pyruvate, phosphoenol pyruvate, will not be converted into pyruvate. In this way, glycolysis will stop when glucagon is coming. So only insulin can activate this pathway. So now regulation of glucokinase activity by glucokinase regulatory protein. So glucokinase regulatory protein, it binds with glucokinase, and in this way it enables it to come there and activate this pathway. Now this is the same when we see that glucagon, it inactivates this pathway, phosphokinase into that, and insulin, it activates protein kinase, and in this way it inactive phosphofructokinase ko it will convert into active phosphofructokinase, and in this way it will show the positive response and fructose 1,6 biphosphate will be formed. So high insulin or glucagon ratio causes decreased cyclic AMP and reduced level of active phosphokinase. So it will reduce the protein kinase enzyme, and this protein kinase enzyme is reduced, so that will decrease the protein kinase A activity, and it will favor the phosphorylation of phosphofructokinase, and this phosphofructokinase is dephosphorylated, is inactive, and hence it will become inactive and favor the formation of fructose 2,6 biphosphate. And then this elevated concentration of fructose 2,6 biphosphate, it activates phosphofructokinase, and this leads to the increased rate of glycolysis. Now we come to the one more pathway which is connected to this glycolysis, that is Rapoport Luebering shunt. So why this pathway is important to join this here? So we all know that red blood cells are unique as compared to other cells. Mature RBC or erythrocyte cells do not possess nucleus nor cytoplasmic subcellular structures. So metabolically mature erythrocytes entirely depend on glucose for its energy. That is glycolysis. So all the RBC cells are dependent on glycolysis. So glucose is freely permeable to erythrocytes. Glucose ends with formation of pyruvic acid or lactic acid, whether oxygen is available or not. The enzyme pyruvate dehydrogenase is absent. Hence the pyruvic acid is not converted to acetyl coenzyme A. Here, that's why the mature erythrocyte, these all changes takes place. So this is the cycle in which the glyceraldehyde 3-phosphate is converted into 1,3-biphosphogycerate. But instead of this 3-phosphoglycerate, here one intermediate is formed, that is 2,3-biphosphoglycerate, and then it is converted into 3-phosphoglycerate. This cycle is known as Rapoport Luebering cycle. In erythrocytes, step 6 of glycolysis is bypassed. This step 6 is bypassed, and biphosphoglycerate mutase converts 1,3-biphosphoglycerate into 2,3-biphosphoglycerate. Then this biphosphoglycerate phosphatase removes the phosphate from to form 3-phosphoglycerate. Then this removes the phosphate and it converts into 3-phosphoglycerate. What is the functional significance of this? The factor which waste energy are not present in red blood cells. Role in hemoglobin. So in adult hemoglobin, when 2,3-biphosphoglycerate concentration is high, it has high affinity to oxygen. But in hemoglobin fetus, 2,3-biphosphoglycerate concentration is low, but affinity to oxygen is more. It is reverse in this. So role in hypoxia. Tissue hypoxia has an important effect on the level of RBC biphosphoglycerate. Inherited enzyme deficiency like hexokinase pyruvate deficiency alters RBC biphosphoglycerate concentrations. So hexokinase deficiency biphosphoglycerate decreases and pyruvate kinase deficiency biphosphoglycerate increases. Okay, so thank you very much. You can also watch the next videos which I will upload on glycogen metabolism and so on. Thank you.